Hi, welcome to the Research Like a Pro with DNA question and answer video for today. The question we're talking about came up in the DNA study group this last week. How do you analyze a hybrid autosegment analysis? And the specific question was talking about why are there some paternal and maternal matches mixed together in one cluster? So we're going to dive into that and talk about how the clustering is done using segments and how we can analyze that. And this is a report that's generated by the third-party tool at Genetic Affairs, and it uses segment data from the companies that provide that. So 23andMe, FamilyTreeDNA, MyHeritage, and GEDmatch. So jumping into how the segments are linked to form clusters, the very first thing that's done is segment linking. And this is from the Genetic Affairs manual, which explains all of the different analyses that you can do at Genetic Affairs. And if you go to page 46, I think it starts on page 45, but the information on this slide is from page 46 and 50 of the manual. So the very first thing that's done is looking at segment linking. And if segments meet the minimum overlap, which you define, so if you want it to be 8 or 10 centimorgans, you can define that when you start the analysis. So if those segments overlap and they meet that minimum that you set, they are then linked together. And this image from the Genetic Affairs manual is showing how those segments are linked. And then these numbers represent how they've formed two segment clusters. So all of these segments are now linked into one segment cluster. And then these three segments are linked into another segment cluster. So that's the second thing that happens is after all of the linking is done and these people all overlap, then when a bunch of people overlap with each other, that's called a segment cluster. And this is a group of matches who overlap on a specific chromosome and location. So chromosome 11 or chromosome 12 and start point XYZ, end point XYZ. And if they overlap there with the minimum that you've set, then they're put into a group. And so if you have a match with 3 segments, that match will be in 3 segment clusters, one for each segment. So matches with more than one segment will be in multiple segment clusters. So these are just groups of matches based on one of their segments and how they overlap with others. After the segment clustering is done, then the tool AutoSegment, it puts the DNA matches into just one main cluster or DNA match cluster. That's usually how it's referred to in the manual, either main cluster or DNA match cluster. And what it does is looks at how many links a match has to other matches in segment clusters. So some people just have one segment, and so they're going to be linked to the people in that segment cluster. But for people who are in multiple of these segment clusters and overlapping with different people and so forth, then it looks at how many links they have to different people through those segment clusters. And if somebody has a lot of links to the same person because they're overlapping on several segments, then they get put into a main cluster together. Now, the trick to know is that even though they're overlapping on a chromosome like chromosome 11 or something like that, the segment data that you're using for this analysis from the company is flat. It's not triangulated. And so it doesn't know, that spreadsheet doesn't say if it's on the maternal chromosome or paternal chromosome. It just knows it's on chromosome 11 or 12 or whatever it is. And so you're going to have some segments that are maternal and some that are paternal. So even within this segment cluster right here, maybe 2, 1, and 4 are overlapping on the maternal chromosome, and 3, 6, and 5, and 7 could be overlapping on the paternal chromosome. But the way that you figure that out isn't by looking at the start and stop points in the chromosome number, it's by looking at shared matches with known paternal cousins and shared matches with known maternal cousins. And shared matches are not used in the AutoSegment report, just segment data is used. So it's up to you to figure out if they're maternal or paternal. So now that we see how it works as far as linking the segments, putting them into a segment cluster and then putting matches into a main cluster. Let's see what it looks like. So in the Hybrid AutoSegment Excel file, which is what I prefer to use, it does give you an HTML file to look at, but mine are usually too large and take up too much memory in the browser. So I usually use the Excel file. And there's two sheets at the bottom of this Excel file. The first one is the main clusters. They're just labeled AutoSegment clusters. And the other tab is called AutoSegment segment clusters. And so this is where you'll find the groups of people who overlap on a specific chromosome and segment. This is the main cluster where people who are linked together with multiple links and segment clusters are put into one group. This is what it looks like if you go to the next tab to see the segment clusters. And you'll see that each segment cluster is given an ID. So this group right here that we're looking at, and these names are anonymized, these are all on chromosome number 1, and the start and end locations are here, and they're all part of this segment cluster number 326. And they also all happen to be in the main cluster number 20. But you can see down here that some of the people in main cluster 163 and 26 are in segment cluster 231 because they all overlap on chromosome 1 at this start and stop location, but they were put into different main clusters. So this is kind of what the report will look like. And now, how do we analyze these? So let's zoom in closer on the first tab. This is the main cluster, and it looks similar to the regular auto cluster matrix reports that you've seen generated before using shared matches, only these ones are used with segment data. This particular auto segment report is a hybrid auto segment, as you can see in the title, and it just means that it combines segments from more than one company. This one has FamilyTreeDNA, MyHeritage, and GEDmatch, and not 23andMe. I didn't have a 23andMe for this test sticker. So what you can do here is look at all of the names, which I've blurred, and see, what testing company they're in. BTC stands for DNA testing company. You can see how much total shared centimorgans they share with the test taker, what main cluster they're in. So the first one is 1, and then it goes down diagonally to the right, and then the next cluster is 2, and then 3 and 4 and so forth. Then in the tree column, it tells you how many people are in their tree, and it's linked to the tree. So if you click on this person's tree count, how many people, you will open it in a new browser tab. Then you can do pedigree triangulation with these matches and try to see if you can find common ancestors among them. Now, if you go to the other tab, the AutoSegment segment clusters, which are overlapping segments just on one cluster, you'll see the names of the people and the total centimorgans, the segment, the chromosome, the start and end location, the centimorgans, the SNPs, the cluster ID, and I took out the DNA match ID so that it would be private. And so this will help you see all the people here that you want to analyze. So looking at this, we don't know which ones of these people are maternal or paternal. Like I mentioned before, we just know that they're matching on one of the copies of chromosome 1 at the start and end location. So how do we figure out what to do next? Well, we just need to review that there are 2 copies of each chromosome. We have 23 pairs. And so we have a total of 46 chromosomes. And at each paired chromosome, one is maternal and one is paternal. And the segment data doesn't say which matches are maternal and which are paternal. So they just get a list of nucleotides from this chromosome and a list of nucleotides from that chromosome, and they're just together in a list, and we don't know which one is which. So we have to do that work ourselves. Although it doesn't say in the segment data, there are some testing companies that have tools to help you assign maternal and paternal. MyHeritage doesn't have anything really, but you can look at the shared matches and then apply labels to help you figure it out. You can also look at the triangulation data when you look at the shared match list that can help you figure out which matches triangulate. And if you don't know if it's maternal or paternal, you can at least say that these two matches triangulate with each other, helping you know they're on the same side. FamilyTreeDNA actually does have the maternal and paternal bucketing. It's called family matching. And so when you add known matches to your tree, then each match who also shares segments with that known match, they are assigned into the maternal or paternal buckets. And that information does come through in the segment data sheet that you download from FamilyTreeDNA. So in the AutoSegment analysis, you can see an M or a P for maternal and paternal for matches from FamilyTreeDNA where you've done that. So that's really valuable to do before generating this report. If you've done 23andMe and you've had at least one parent tested, then you will have the maternal and paternal labels on all of your matches, and that can be really valuable. Otherwise, you won't have that. And then at GEDmatch, when you submit files from GEDmatch to the Hybrid AutoSegment report at Genetic Affairs, it asks you to submit the segment data and the triangulated data so that it will only put GEDmatch matches who triangulate in the same cluster. So it really helps you figure out where people belong as far as which cluster they should go into. Okay, so now that we've covered that, now we know that we need to add a column to analyze our segment clusters to show us whether or not a person is a maternal or a paternal match. So I just inserted a column into my Excel spreadsheet for me to annotate whether that person is a maternal M or paternal P match. And I just went down my list and went to MyHeritage. I opened up matches at FamilyTreeDNA and GEDmatch and typed in the names and looked at shared matches and my notes to see if I had identified any of these. And some of them I couldn't identify. That's where they're blank. But this first one I could see through shared matches that it was a maternal person. And through shared matches, I could also see that some of these other ones were probably paternal. The only one that I knew for sure was this Carol Hansen, and I actually found this cluster 132 because I was doing a control find in the Excel spreadsheet for Carol's name so that I could analyze clusters that had Carol in it because I'm looking for more matches who share DNA with Carol. And so she's in cluster 156 and she's paternal. And so some of these other paternal matches are the ones that I'm now interested in because she's paternal. I'm looking for a paternal ancestor for my testing group. So once I've figured that out, now I can go forward and do more analysis on these matches who I've deemed relevant. One thing that I have noticed is that often people who are linked in the same main cluster, like all these people who are 37, are probably related on the same side of the family. And that's just anecdotal evidence so far, but they're sharing different segments. And so they're probably all going to be paternal in this cluster 37. So it's helpful to have both segment cluster numbers, the main cluster for the DNA match, and then the segment clusters grouped by overlapping segments. So the takeaways are that Hybrid AutoSegment gives you two types of cluster: the segment cluster, which is people who overlap on a specific chromosome and location, and then the main cluster, which is the DNA matches cluster. And that's people who are linked by one or more segment cluster. And then just remember that maternal and paternal matches are going to be mixed together in those segment clusters. And so it's helpful to add a column to the Excel spreadsheet and annotate with maternal and paternal. And as you find matches who overlap and are also on the same side, then you can go ahead and analyze the tree for common ancestors. So hopefully that helps with using the AutoSegment tool. And if you haven't tried it yet, then I encourage you to add your notes about who's maternal and paternal, and then try it out and see if it can help you find more matches that are relevant to your project. Thanks.